All right, so the grand finale is, how do cells sense time? How do they know when it's time to grow? This loops back into the insulin part. But the thing is, when cells grow, as they grow, they need a clock. Because growing takes a lot of time. If you're building a house, you got to get all the materials together. Then you got to put up the walls first, and then you got to put on the roof, you know? Cell is the same kind of thing. There's a lot of things that have to be built, put together, and put together in the right order. So that means a cell can't just divide like that. A human cell is going to take about 24 hours to divide, going as fast as it can. And it needs a clock to keep the proteins that don't need to come out yet, to hold them back, and to keep the proteins that need to do something to bring them out. And also, we need to be very tightly regulated. You want to grow a lot when you're little. You don't want to keep growing at the same rate your whole life. At some point, you stay about the same. And the thing is, cells are growing, cells are dying, so you keep that going on. But you need to tightly regulate it so that they only grow when they need to. Once it starts, you need to have the timing. And what regulates this is, probably as you guessed it, kinases and phosphatases. Kinases tend to turn these processes on. Phosphatases tend to turn them off. Each step has its own little army of dedicated kinases that start it. And so that means a cancer researcher working on drugs to interfere with this process will be working with a lot of phosphate-related molecules, because that's what the cell uses. Now, these proteins, they have to go throughout the entire cell. You know, growth involves the whole cell, and they have to respond to time and internal checkpoints. So there's not really a first messenger here, unless you want to consider insulin or something like that as being the first messenger. This is what the cell cycle looks like. And if you go work at the Hutch, you probably will see this all over the place. And you see how you add this up. It's about 24 hours for it to go through the 4 different phases. Each phase takes its own time and has its own steps that it has to go through. And you can't start step 2 until you're done with step 1. You know, that's how the phases got their definition. So keeping all those molecules in order, if you think herding cats is a bad metaphor, you know, is easy to do or something like that, then think about herding proteins. You know, you've got like 10,000 proteins, and they're all floating around in solution, right? You've got to hold these ones back. You got to get these ones over here. It's like the drama production from hell or something like that. You've got to get everyone on stage at the right time. So I have a lot of respect for people who manage, because the cell manages this kind of movement. But needless to say, there's just, oh, and by the way, there is this G0 phase, which just means the cell is resting and not growing. These are the 4 phases of cell growth. When you have these 4 phases, you have different kinases that are active for different phases. So here you have CDK2, the K means kinase, and you see that it is active at the beginning of S phase. CDK2 binding a different protein, this is bound to cyclin E. When it binds to cyclin A, it remains active, but it will initiate the G2 phase. And then you have a different kinase, CDK1, that's associated with the actual mitosis stage. Different kinases, or different kinase protein complexes, which amounts to the same thing, they're going to be turned on during different stages. And so if you see a cell that is not able to turn itself off, maybe it has a mutation in one of these things. And in general, kinases will turn stages on. You can see that these are happening near the beginning of the stage. And phosphatases will turn them off eventually. Phosphatases can be what's bringing the kinase activity down. Very broad brush. So cell growth can be a complex set of kinetic equations, and we actually are getting pretty close to modeling a rudimentary model of all these equations together. When something goes wrong with all of these K values and all of these enzyme interfaces, the cell can keep going in the circle. It doesn't matter if it's going a little slower. What matters is that it's not stopping, and it keeps growing, and then you have a tumor. Remember that growth factors turn on these pathways. So if you have a mutation in this pathway, and remember that we have a lot of off switches. If one of those off switches is messed up, let's say that the residue that's supposed to hydrolyze the GTP and turn off the G protein, what if that's mutated? Well, then it's not going to be able to turn off very well. And then you'll have unregulated growth. So let's say that you have a kinase that is mutated so that it's always on. That would be an oncogene. And then there are other cells that automatically turn things off. And when those are mutated and those are out of the way, the stuff isn't turned off anymore. That would be a tumor suppressor gene. When it normally suppresses tumors, you mutate it away, and you aren't activating it, you're messing it up. A tumor suppressor, getting rid of a tumor suppressor can be as bad as activating a kinase. I believe the RAS mutants that we were dealing with were constitutively active RASs, which would keep the cell growing, and that's what you saw in the Wells paper. So here's just an example of oncogenes in green and tumor suppressors in pink. Notice that you see a lot of Ks in the oncogenes, and that's all I'm pointing out. This is not a hard and fast rule. This is just me noticing, boy, there's a lot of kinases there, you know, and things like that. Cyclin, by the way, comes from cell cycle. But this means that underneath superficial differences in the starting in different tissues, different cancers might not be all that different after all. And so you'll struggle with this as you read the Emperor of All Maladies, because this is really a question. Does it make sense to have a cure for cancer? Well, yes and no. From this standpoint, it actually makes more sense because signaling pathways, different signaling pathways can be messed up. But people have been trying this for a long time, and they've been finding out that cancers are very different. In fact, it's gone so far that most people were so convinced that cancers were so different, it's kind of a surprise to come around and find, oh, some cancers are pretty much the same. And in fact, this seems to be a big movement. There are tissue agnostic treatments now, where you can have, so you have like 3 different types of lung cancer, 3 different types of liver cancer, and 3 different types of colon cancer. Sometimes you have the same mutation, mutation B, that will cause lung cancer in a lung cell and colon cancer in a colon cell. The same inhibitor, or the same drug that targets that mutation, would work for both of those if you got it in the right place at the right time. And so because signaling pathways are the same, there is the option for this to be the case. Everybody was really keen on this when they first found the pathways. Then everybody soured on it. And now we're kind of in the 3rd stage of, well, maybe it works sometimes. And now that we're able to look at, like, this actually has a big impact because traditionally you have to get all people with the same kind of cancer for your clinical trial. But if you have a case like this, you can get all people with the same kind of mutation. And you can put them in your clinical trial, and it actually can help clinical trials for rarer diseases to go on because you find out different people have the same disease. This is a whole story about it, so I'm going to let you look at it a little bit more if you're interested. But the main thing I want to point out is, right here, notice the K. You're looking through a story about cancer. You see something. It doesn't explain that K at all. But I was like, I'm going to do an experiment. I'm going to make a prediction that that TRK means kinase, because kinases are like this. And so I really did not know for sure, but I looked it up, and because I'm showing it to you, you can probably guess that I was right. I wouldn't tell you if I was wrong. But the thing is, there's this kinase that is active in mutated in different forms of cancer. And I looked it up. It's actually hard to find in the medical literature because they're always like, everyone's so scared of biochemistry. They don't want to say the word kinase. But I eventually found out that there's these TRK proteins, and as far as I can tell, that they are actually kinases, okay? The TRK family of proteins, and they send extracellular signals to the nucleus, and they're involved with these famous kinase pathways. If you mutate them, you have these different types of cancer, whatever tissue it's in. And so this drug company is going after these. Oh yeah, it took forever to find this, and I hope I'm right. But that's the main thing is when you see K and you see cancer, you probably think kinase. Emperor of All Maladies will have a lot more information on this. Hold on to some of this. Hold on to some of this until the quiz 2, because I'm going to ask you some big picture questions when it comes to quiz 2 on this stuff. I'm just showing this to you to say, look what you don't have to do, okay? If you really want to look at the linear problems, maybe that would actually be one way to study, but I will just leave it at that, all right? Thank you for sticking around. I know it's a long day, but it's a long chapter, and it's an important chapter, and I'm really glad we were able to go through it. Enjoy Friday off. I will see you next Wednesday. And I actually haven't looked ahead to what next Wednesday holds, so I'll send out an email, but I believe we're going to be doing presentations. Is that right? So next Wednesday presentations, get ready and let me know if you have questions. All right. Happy Easter, everyone.